Hi everyone. Thanks for joining in today. Today we'll be talking about microbial genetics. We'll try to understand how translation happen both in eukaryotes and prokaryotes. So without further ado, I'll just read out the learning objectives for today. So we'll compare and contrast key features of eukaryotic and bacterial chromosomes. We summarize the steps of bacterial DNA replication and the enzymes used in the process. We also summarize the steps of bacterial transcription and define key factors being used. We also summarize the steps of bacterial translation and define the key factors used. We will identify differences in bacteria versus eukaryotes for DNA replication, transcription, and translation. And lastly, we will define operon, which is basically, okay, we'll talk more about it. We will define operons and differentiate between repressors and inducible operons and how they work. So, in microbial genetics, today we are only focusing on eukaryote and prokaryote. We are not going to talk about viruses and prions. Um, these are just we are just focusing on cellular organisms. So the focus will be on processes in both bacteria and in bacteria stroke archaea, which are prokaryote, and compare and contrast them with that of what happens in eukaryotes. So for DNA replication and all of this I'm talking about to happen, you need a set of something which we call a chromosome. Basically, these are a discrete structure, cellular structure composed of neatly packaged DNA molecule because you have to move from DNA to RNA, then to protein, and throughout all of these cellular processes. So now let's compare eukaryotic chromosome to bacterial chromosome. I'm sorry, bacterial and archaea chromosome. So basically, with eukaryotic chromosome is located because eukaryotes have a nucleus, so their chromosome is present in the nucleus and it varies in a few number from probably a few numbers to hundreds, and then they are linear and can occur in pairs, so they can come as diploids or synchro, which is haploids. So you can think of it in the relationship with mitosis and meiosis. And the chromosome of eukaryotes are mostly larger. They are actually larger than the bacterial chromosomes. So, and as we all know, bacteria doesn't have a nucleus. For that reason, their chromosome should be is present in this cytoplasm, and they usually have only one, and then typically it's circular in shape. And then we have they are actually single copies and then smaller. So they have single copies of their chromosome, they are smaller, and they have to replicate it when anytime they have to replicate it again anytime they want to like form more of themselves. So how variable are chromosomes in size? They actually are hugely variable. So for example, if you pick E. coli, the bacteria have been working a lot in the lab, um, it have a single actually have the bacteria have a single chromosome, but the size itself ranges from 4.3 million base pairs, coding about 4,300 genes in just a single chromosome. And from here, you can see it's just 1 millimeter long in length, as you can see just rightly from here. This is when the whole chromosome have been unwind and being stretched along. So just imagine how smaller it will be when it's circular. Comparing that to tapeworms, you can clearly see that the tapeworm chromosome is actually larger. That is 115 to 141 mbps, encoding about 10 to 12,000 genes. And it's actually having more of a multiple chromosome, usually 8 to 9. So you can realize how variable chromosomes can be depending on which organism they are being found. So to describe the whole process, we have to understand that there is actually four major processes involved. We have the DNA replication, where DNA have to be made doubles, and we have DNA transcription, where DNA have to be transcribed into mRNA, And then we have the last stage, which is protein synthesis, where the mRNA have to be translated into proteins. And the last one, we will talk of options, where which is basically different systems of what is happening in these prokaryotes. So back to our friendly friend bacteria. So as we talked about earlier on, bacteria doesn't have a nucleus, so their chromosome is present only in the cytoplasm. So then how does the DNA replication happen in these bacteria? It's happening through binary fusion. As you can see from here, this is a single bacteria, young cell, happy is eating, and then it realizes it have to make more copies of itself. So what does it do? It first make a copy exactly, it makes exactly the same copy of the this chromosome here. So here you can see it's making two copies of them, and then basically there is a protein band that forms in the center of the cell just to separate the two chromosomes. Is just replicated of itself. Not only is the chromosome being replicated, but any other thing being present is being replicated. And here there is basically a formation of septum which begins, and finally septum is completed, and then you have two daughter cells or two new cells of bacteria. This process is what we call binary fission. So, as I have described earlier on, in bacteria DNA replication, it's basically making two copies of itself. So we have a semi-conjugative replication. Basically, each daughter molecule is identical to the parent in composition. So as I said, you make copies from this very same thing, and you are making the exact copy. So you are going to have the same thing, which is termed as a semi-conjugative, a semi-conservative replication. So in these points, you can see there's a template strand, which is the original one. And here replication is happening from two places. So we have from the two forks. It can happen from the first one or the second one. So replication is happening at both forks, and then you end up having two identical genome or chromosome. And then lastly, the chromosome is being separated into two daughter cells. So that is basically how the semi-conjugative replication happens. And I want to point this thing. You can clearly see that there is a bi-directional origin of replication. Replication is not happening on one side. So at each replication fork here, so one is here and one is here, both of them, replication is happening on both sides at the same time. That is termed as a bi-directional replication. So take note of that. Um, how do I play this? Okay.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
Okay, so that was a short video of how DNA replication occurs. And just to reiterate what we saw, So DNA, during DNA replication, the parent DNA is being split into two, and we have the leading strand and the lagging strand. Pay attention that replication only happens in the direction of 5, 2, 3, and basically the next available nucleotide, for example, let's say it's adding from this direction, the next available nucleotide here. So the next available nucleotide will be only be added to the OH group of the leading strand or the lagging strand, basically based on where the polymerase is present. But as you can see, replication is happening on both strands at the same time, on both the leading strand and the lagging strand. The only difference is that replication happening on the leading strand is more continuous. There's no stop on it, or basically there's no breaks on it. However, what is happening in the lagging strand is that um, when the DNA polymerase attaches to it, it attaches this RNA template basically on the lagging strand, and then it can start growing on it. But as you can see, polymerase attaches the RNA template and it start adding the nucleotides on it. It can add totally like throughout continuously like the leading strand. So you can see ahead in front of it, there's also another um, attachment of another RNA primer, and there's also another replication being beginning. So basically, you don't have the continuous one, but you have these short fragments which keep happening on the lagging strand. So, from the video and how I've explained the whole thing from watching the video and also the explanation I've given so far, we've now understood how replication is happening. However, let's compare to how replication is going to happen also in eukaryotes. So in prokaryotes, we've understand there's a leading strand, there's lagging strand, leading strand goes continuously, lagging strand, there's a bit of a breaks and multiple attachment of RNA primers. So in eukaryotes, the DNA replication is actually similar to that of the bacteria and hacker DNA. However, because there's no nucleus, this is only going to happen within the cytoplasm, and then replication will proceed from multiple origins. So as you can see, in the eukaryote, we only showed only one point where the leading strand and lagging strand was happening. But here, as you can see, just on one DNA strand or on one parent DNA, you can see there's one bubble here forming both leading and lagging strand, and there's another replication origin of replication happening. All of them are happening simultaneously, and at the end of the day, they all transcribe into a whole new parent DNA, as you can see from here. I want us to watch a short YouTube video on this. So, um, okay, stop sharing screen. Then let me try and share the, okay. Mm, just a minute. I'm trying to navigate this whole thing. Okay. So that I can share the video for us to watch. Okay. I hope you guys can see my screen. So let me press play so that we can watch this short YouTube video.
The ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
Okay, so now that we've finished watching this video, we can now go back to our slide and then we can continue. So from the video, it talked about how the lagging strand, what is happening on the lagging strand, and basically talking about how the telomeres comes in and then closes the gap which is being formed because there is a lot of multiple priming on the three strand. The lagging, there's multiple priming on the lagging strand during replication. So in both eukaryotes and prokaryotes, there are different things. There are different polymerases involved. As we've learned earlier from the prokaryote, we have polymerase like DNA polymerase 3, which is happening in the replication. What is happening in eukaryotes is that in eukaryotes, there's a requirement for both polymerase delta and epsin, as I've listed here. So they both perform the different functions. So polymerase epsilon will be on the leading strand and polymerase delta will be on the lagging strand, and both performing similar function as what polymerase 3 will do in the case of prokaryotes. So here it's a practice for us, and like how we normally do, we will tackle this when we meet in class and we can discuss it among ourselves and have fun. But you can practice this on your own. So now we've talked about DNA replication and we've understood how DNA is being making into multiple copies. Now, now it's for time for us to talk about DNA transcription. Basically, where the DNA which has been made, the copy which has been made is being turned into mRNA. So gene expression involves the process where genes which are encoding for proteins are being transcribed. And basically, this whole process involves two steps. We've talked of DNA to mRNA and then from mRNA to protein. So imagine this as oh, I didn't know my pointer wasn't showing. Okay, here. So imagine this. So now we have our DNA strand here, and then replication happens. So then we end up having a template strand which we just created from this parent strand. So then this template strand will now be changed into mRNA, and then we have transfer RNAs which will come and basically attach to this mRNA, and then the transfer strand will come transcribe it into protein by coming together. The transfer RNA will be coming together with the appropriate amino acid coding for this, and then this amino acid will then be attaching, will form bonds to the nearest amino acid based on what the transfer RNAs are coding from this mRNA, and then they'll keep attaching to each other, and at the end they'll form a protein. So as you can see, basically from here, you can see that basically the same amino acids, this same amino acid are having different transfer RNA anticodons. So even though they are all both threonine, they have basically, let's try and read this mRNA. So ACU and ACG, even though they are different, they are both coding for threonine, and it happens mostly. There's a lot of amino acid which are actually like this, so we have to pay attention to this. So now let's dig into this whole process of from DNA to mRNA. It involves three processes. So we have the initiation, elongation, and termination. So initiation basically means the attachment of the whole this polymerase involved, the RNA polymerase involved, and then elongation, just similar trend as if it's happening DNA replication, and then lastly termination will happen. So let's watch this video.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
All right. So then, just to reiterate what we just saw from the video. So now we have DNA replication happening. We now have the replicated DNA that we have. So we have to change it to mRNA. So basically, the mRNA polymerase attaches to the promoter region of the template strand that we've made, and then the RNA polymerase initiates this. It recognizes the initiation codon and basically keep adding of the RNA nucleotides complementary to what the DNA sequence is coding for. So, for example, if it's gene is gonna add C, if it's A is gonna add T, just doing the same thing over and over. And then, so as this whole mRNA transcription is happening, what I want you to know is that remember that uracil, so uracil is being placed as adenosine complement. So in DNA, it would have been like A to T, but in RNA, it's going to be T to a U instead of a T to a A, because uracil is being replaced with an adenine. So this whole transcription, or sorry, this whole RNA transcription is going to happen. So it keeps going. This is the polymerase. It will keep going and keep transcribing till the whole RNA has been completed. And then eventually it terminates. That's when it reaches it and it reads the stop codon. So for it to start, it needed the initial start codon, which is ATG or AUG. depending on whether you are saying it in the RNA format or the DNA format. So let me just stick to one. So in the RNA format, should have in the DNA format, it should recognize ATG, and then it keeps transcribing till it sees the stop codon, then it stops. Stop codon can be TGA, it can be it can be also be ATG. So please practice this. So now we've understood the whole process of DNA replication. We've now understood of changing of the DNA also into mRNA using RNA polymerase. Then lastly, we also have to discuss how the mRNA is being translated into protein. So this whole process also involves three. This process will involve also a three-step. We will also have an initiation, elongation, and a termination. Mind you, this will be done by ribosomes and transfer RNAs. Basically, they'll be playing a role in synthesizing the mRNA into proteins.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay. So as what we've seen from the video, it becomes super simple. Over here are just some key terms which summarizes the whole translation process. So we have ribosomes, which consist of both larger unit and smaller subunit. And then within the ribosome itself, there is an A site, basically the first site where the transfer RNA will come and sit. There is a P site and there is an E site where the transfer RNA will exit. So from the video, transfer RNA comes and sits at the A site. Transfer RNA is already coming with it codon with it amino acid that codes for the anticodon. And then the anticodon recognizes the RNA codons, and then it binds to it, or basically it attaches to it in the ribosome, and then it's being the whole attachment of the anticodon and the mRNA is being moved to the P site, and then it then goes. So when it moves to the P site, the amino acid already present on the E side is being added or being binds to what is on the P side, and then the ones in the E side then exist. So basically, the anticodon once the transfer mRNA, or sorry, once the transfer RNA moves to the exit or the E side, its coding amino acid is added to the amino acid on that of the P side, and then the empty the empty transfer RNA is being exit in the exit site. And then I've already explained the whole thing. Yeah, basically, so these are what you need to know in order to fully understand this. So just to go over again, the A side is there, the entrance or entrance site for the transfer RNAs. Transfer RNA binds to the codon on the mRNA. And then in step 2, there is the formation of peptide bonds, which is going to happen from the amino acid in the A side and the P side. And then 3 is just the discharge of the transfer RNA. So basically, once this bond is formed between the transfer RNA on the P side and the A side, the transfer RNA in the P side will be moved to the exit, which is the E side, and then the transfer RNA goes, but the amino acid still stays. Basically, it's just forming the bond with the other amino acid in the A side. And then the 4 is just basically their first translocation, where another transfer RNA also comes in. And then in 5, it's also the newly coming transfer RNA also form the bond with the already formed amino acid complex already here, and then it keeps adding to it, and then the next one also exits. So this whole process keeps happening, repeating itself. New transfer RNA comes, it comes with an amino acid, it's add the amino acid to the already existing chain of amino acid, then that amino acid goes off, and it keeps repeating till the whole thing finish. This slide I won't go through. This slide contains exactly the whole process that we've watched from the video and what I've also described. So now let's understand the difference between the whole transcription process, transcription and translation process happening in both eukaryotes and then prokaryotes. And as you can see, the whole difference happens in the location. So bacterial cell is going to happen in the cytoplasm because there's no nucleus. And then in eukaryotic cell, it's going to happen, the transcription and the RNA is first going to happen. Transcription and RNA process is first going to happen in the nucleus. And then the RNA will be processed into mRNA, and then the mRNA will now move into the cytoplasm where translation will happen, basically turning the mRNA into polypeptide chains or protein. Second point to take note is that there is co-transcriptional there is a whole co-transcriptional translation only happening within the prokaryote. So as you realize that okay, I think from the previous slides, the whole thing happens only within the cytoplasm of the bacterial cell, but in eukaryote, both is happening only within the nucleus and then before it's being moved out. And this is just basically a picture of what is happening. In the bacterial cell, you have basically transcription happening here, and you are producing more of the RNA. Basically, this is the RNA polymerase transcribe more of mRNA, and just right away just shoves this into the chromosome because everything is right in the cytoplasm. So the transcription happening in bacterial cell only results in an mRNA, but the one happening in eukaryote will first result in a premature mRNA, and then the premature mRNA have to be processed into a mature mRNA before it exits the nucleus into the cytoplasm for transcription protein translation to happen. And as this is going on, first in the eukaryotic cell, when the DNA is being transcribed into premature, when the DNA is transcription occurs and it's being transcribed into premature mRNA, there is two things that are being coded. So in the eukaryotic cell, there'll be a code for introns and exons, and then the introns and exons, there have to be a splicing that have to occur basically to splice out all of the introns so that the exon can code for what it needs to code. So now let's pay attention here. So this is just a classic example of what I was talking about. This is a DNA template which have both a nice arrangement of an intron, of an exon, intron, exon, intron, exon, intron. And something that you have to pay attention is that introns are there's the interval sequences that do not code for any protein. They don't code for any protein, they are just present. And then the exons are just the coding regions. This is happening in this is only happening in eukaryotic cell. Know that in the bacterial cell, this doesn't happen. So this is a classic difference between bacterial cell and transcription and translation happening in bacterial cell and that of eukaryote. So back to this slide again. So we have a nice arrangement of exons, introns, exons, introns. So now we've just transcribed everything from the DNA template. We form our mRNA. And then from the mRNA, we have spliceosomes. So spliceosomes are just enzymes that comes in and just cut or clave off the introns so that at the end, you'll be having a nice arrangement of exons, which are basically the coding regions of the whole transcribed DNA. And then the exon, the spliced mRNA is now sent out of the nucleus for the whole transcription, protein transcription to happen. So that is just a clear difference of what happens in the bacterial cell and eukaryote. This slide is all for you to practice. So take some time, practice. We can also go again over it again in class so that we can all discuss. So now we've discussed DNA replication in both prokaryotes and eukaryotes. We've talked about transcribing DNA to mRNA and then translating it into proteins. So now let's talk about operons, basically regulation of gene expression in bacteria. So operons basically are a coordinated set of genes that regulate as a single unit in bacteria and archaea, but not in eukaryote. So just pay attention to this. So this is just a whole gene of a whole genome of a bacteria, just as a cartoon. So as you can see from here, there's this region called the regulatory region. So this region encodes for a repressor protein, basically a protein which is going to repress the transcription of this whole genome region. And then here we have a promoter region or a promoter. Basically, this is responsible for promoting the gene expression. And then this part codes for a promoter to promote the gene expression. And we have the operator. So this operator region is just present for the binding of all the repressors. So the repressor have to locate the operator and just repress the transcription of the gene. So this whole process, this whole area from this promoter region to the rest of all the downstream genes are called the operon. So now let's describe how this whole operon work. So we have the regulatory gene here, which encodes for the repressor protein. Then the repressor protein will bind to the operator site and blocks mRNA transcription because blocks the RNA polymerase transcription because RNA polymerase will only recognize this promoter region. So RNA polymerase is supposed to bind to the promoter region and help the transcription of all of these genes, in this case A, B, C, D. But because we have a promoter region present here, the promoter region will code for a gene, and that gene will produce a protein that will come and binds to this operator region, halting or blocking the transcription of RNA polymerase. I hope this is clear. We'll go deep into it in the next few slides. And as you can see, just one RNA polymerase is going to transcribe all of these downstream genes just once, producing different proteins, as you can see from here. So now let's talk about how operons work. So we have two types. We have inducible operons and we have repressible operons. So inducible operons are they work in this manner. The operons are always turned on by substrate for which structural genes encode. So that's basically what I explained earlier. You code for a certain the operate sorry the regulatory region codes for certain proteins and then it just activates it. So enzymes are needed to metabolize the nutrient only produced when the nutrient is present. So for example, if a specific substrate is responsible for activating some enzymes which are present for the activation of the whole gene, that substrate or that enzyme have to be turned on or have to be present before the whole transcription happens. If they are not there, the whole transcription of the genome won't happen. I'll show you an example of what an inducible um, operon, how an inducible operon works. So basically, just imagine it as your light switch and electricity. You have to turn on your electricity. So the enzyme have to be present to turn on transcription. So you have to go to your wall and turn on your light before you can, have the whole light in your room. So basically, that's how the inducible operon works. Now, the opposite side is repressible operon, and this happens in genes which have in genes which is already turned off. So the product is already turned off, they are naturally off, and then you need the enzyme basically to basically activate the whole transcription process or prevent what to get rid of whatever is preventing the gene transcription from happening. So that is just the opposite. So in the inducible one, you need a certain enzyme or substrate to be present for you to have gene transcription. And in the repressible one, gene transcription is already hot or turned off, and you need a substrate to activate it back. So now let's talk of how this thing works. So first, we are focusing on the inducible operon. Inducible, yeah, operon. So here, we have a normal genome where we have the regulator, we have the promoter where the RNA polymerase will bind, and we have the operator where the repressor protein is. So in the inducible one, in its nature, the regulator protein, the regulatory gene codes for the repressor protein, which binds to the operator. So in nature, it has bind to it, and there is no transcription of downstream genes. And then, even though the RNA polymerase is still present, there's no transcription happening. And then we have the inducer, which is basically the substrate which is going to be produced. So so basically we will need the inducer or the enzyme or the substrate that is going to induce the transcription to happen. So even though here I'm iterating again, even though here the polymerase is present, but it's being blocked by the repressor, once we induce it with the presence of, for example, lactose, when lactose comes in, lactose get rid of the repressor protein here, and then the RNA polymerase will now have the ability to transcribe the downstream genes. So without the presence of lactose, transcription will never happen. But when lactose comes in, it get rid of the repressor here, and then the RNA polymerase can work. So then, as I explained earlier, now we've gotten rid of um, the the lactose binds to this pocket on the inducer, on the inhibitor or the repressor. Lactose binds to this pocket on the repressor, and then it takes off the repressor, and then RNA polymerase can go ahead and transcribe all of the genes at once. So, as I told you, you are going to make different proteins all at once, just straightforward. And then after transcript, the whole mRNA transcription is done. Lactose get off from the binding pocket here. Lactose goes off, and then the repressor comes back and attaches to the operator region one more time. And then, even though polymerase is still present, there's no transcription happening. So now let's talk of the opposite one. One prime example of the opposite one is the adenine operon. So in the repressible operon, transcription is just happening all the time. So as you can see, polymerase is there. There's no repressor present on the operator side. So transcription is happening. Everything is being transcribed. All of the things are being, all of the downstream genes are being produced. And then When it's time for the transcription to be hot, one of the downstream genes will code for the protein arginine, and then, for example, this protein will code for arginine. Now, arginine comes in, and then arginine recruits this repressor, and then the repressor, basically here, the arginine comes in, binds to the pocket of the repressor, recruiting the repressor and bringing it back to the operator region, basically halting or blocking the whole mRNA transcription process. So I hope this explain the two type of inducible operon. We have the two type of operons. First is the inducible one where you need the substrate to be present for transcription to happen. And then we have the repressible one where you need a subscript to be present to block transcription. So practice more in your microbial genetics practice book. I noticed some of you have been missing class recently. I'll be marking for attendance on Friday, so you have to be present. If you are not present, I'll put it on file so that later on, if you come in and be like, "Oh, we didn't say this, or the professor didn't show this, or my grade wasn't good, my this wasn't that," then I have an evidence showing you never show up for class. So if you don't understand anything, that is on you. So I hope to see you on Friday, and also remember on Tuesday we have lab both morning and evening. Be present. We also be taking attendance of both. So have a nice evening. And okay, oh, and also there are TA hours on Monday. So because we don't have an in-person class on Monday and Wednesdays, the TAs are present in RC 417. So you can go there and ask them questions. Or now they realize you guys are not coming, so they decided that we've decided that we are going to do an online TA section. So during the same time as in 12:30 to 1:40, there will be a Zoom link where if you have questions, you can just join in and ask for help from the TA. So I'm saying it again, there are TAs hours happening on Monday and Wednesday, and then Friday we have the in-person class. So if you have any questions outside of class that you can ask me, or you want to ask the TAs, you can ask them on Mondays and Wednesdays from 12:30 to 1:40. We have TA hours, so take note. All right, have a nice evening and see you on Friday. Bye.